Okay, look, protein synthesis, chapter 8, section 4. Today we're going to illustrate the protein synthesis, chapter 8, 4.4, okay? In the first step, you have to note that protein synthesis includes two steps. Step one called transcription and the second one called translation. Look at the figure initially. During transcription, during transcription, we have a gene on DNA. It consists of two strands. Each gene has a promoter, beginning of the gene, and also it has termination signal, end of the gene. Promoter and termination signal. This is marked the beginning of the gene, and this one marks the end of the gene. First, the gene is copied. Okay? During transcription, first the gene is copied. But be careful, only one strand is copied. The process called transcription. And the copy of the gene, this is the copy of the gene, called RNA. Okay? In the first step, the protein synthesis, which is called transcription, the gene is copied, and the process called transcription, and the copy of the gene called translation. Look, during transcription, for example, we have A, G, T, C, C, A, for example, G, G, A on the DNA, which acts as a template. Okay? During transcription, the enzyme which transcribes the DNA, it adds, instead of A, we have the ZTA, it adds U, because there is no T in DNA, in RNA, and then for the it adds C and T, A, and so on. C, GC, GA, U, and GGA, it will be CCU, because there is no T in the RNA. This process is called transcription. And then the RNA do what? It leaves the nucleus to the cytoplasm. Then, look, each three nucleotides on the RNA, it will be a codon. Codon for what? Okay, codon for what? Look, the proteins consist of a series of amino acids. The amino acids joined together by peptide bond. Each of these amino acids, each of them, it has a specific codon on the RNA. Okay? And three adjacent nucleotides. On the RNA, it will be a codon for a amino acid in the proteins. So during translation, you can say, the RNA acts as a template for making what? And directs the assembly of the amino acids in the proteins. And also we said that three adjacent nucleotides will be a codon for an amino acid, and also, for example, this one, U, for example, it will be a template for this amino acid, and this one, CCC, it will be a template for this one, and so on, okay? So protein synthesis, we have two steps. During transcription, DNA acts as a template for making RNA, and during translation, RNA directs the assembly of the amino acids in the proteins. First one. Second one, this protein, which is made by the transcription or translation, both steps together, may be any proteins in our body. Okay, for example, it may be, it will be a hormone, amino acid-based hormones, or maybe it will be an enzyme, help in digestion, for example. Also, it will be maybe actin and myosin. For complement system proteins, interferons, antibodies, all of them, it will be the protein that produced in the same way. Okay? This is the first step. Second one, you have to know other things for the subject. For example, look, You start at this point, this one. Look, Mr. Figure 11a summarize what the flow of genetic information in the eukaryotics during transcription. During transcription, DNA acts as template for synthesize what? The RNA, and also during translation, the RNA direct the assembly of the proteins. Also, you can see the assembly of amino acids in the proteins, okay? And also be careful during protein synthesis, yeah, you can say protein synthesis based on information in DNA, but carried out by RNA, right? And also this central concept can be symbolized as what? You can say as DNA, the information transmitted to RNA, and then finally to the proteins. And also we said that we have important proteins formed during this way, which are hemoglobins, antibodies, actin and myosin, interferons, complement system proteins, all of them produced in the same way. That's first. Second one, on the other hand, on the other page, we have differences between an RNA and DNA. Okay, we have 4 differences. But first, you have to memorize them, the differences, on this figure. Look, let's compare the DNA and RNA in the number of strands. First, you have to know that DNA, it's a double strand molecule. But look, this first one and this one, the second one. But RNA is only a single strand nucleic acid. Double strand, single strand, first. 2, the DNA nitrogenous bases, we have A, G, C, T. But RNA nitrogenous bases, we have A, G, C, U. We have U instead of T in the RNA. Third one, the sugar in the DNA, called what? Called deoxyribose. But the sugar in the RNA called ribose. Okay? 3. Finally, DNA is very long. Look, it's about, in the human body, in the human cells, it's about 25,000 genes. So it's very long, 100 or even 1,000 genes. But RNA very short. A length of RNA, it's only about the length of a gene. Okay, that's 4 differences between DNA and RNA. Also, if you look at the figure on the next page, you see the difference is very clear. DNA. First, DNA is a double-stranded nucleic acid. This is first one, and this one the second one. But RNA only is a single-stranded nucleic acid. Okay, but sometimes the RNA, the single-strand RNA, be careful, it folds, and during folding it forms short double-strand sections. And also, if you have hydrogen bonds, it will be between in the RNA. It will be between A and U because there's no T, and also it will be between C and G, the triple bonds. So the triple bonds is same in both DNA and RNA. To the sugar in the RNA called ribose, and also the sugar in the DNA called deoxyribose. 3, the RNA nitrogen bases will be AG. CT, but in the DNA it will be AG. CT, finally, it's very long, about 100 or even 1,000 of genes, Balam. This one very short, about a length of 1 gene. That's all difference between them. So the GC triple bond, phosphate groups in the nucleotides, purines A and G, all of them, it will be same in both DNA and the RNA. That's all for the first lesson.